All living organisms evolve by accumulating mutations and by reshuffling genetic information through recombination. Beyond mutation, bacteria also have specific mechanisms to transfer genes between cells, collectively known as horizontal gene transfer processes. The most typical processes of horizontal gene transfer are transformation, where bacteria uptake new DNA from the environment, which is not thought to happen in the mammalian intestine. Then we can have conjugation, where the transfer of DNA by plasmids is accomplished by cell-to-cell contact. Finally, there is transduction, which is a process that relates to the transfer of DNA by bacterial viruses, also known as bacteriophages, phages for short, which are recognized to be the most abundant biological entities on the planet. I am Nelson Frazão, and together with Isabel Gordo, both from the Instituto Gulbenkian de Ciência in Lisbon, Portugal, have written a piece for this issue of BioEssays about the importance of understanding mutation and especially phage-driven horizontal gene transfer during evolution of bacteria in the mammalian intestine. Mutation and phage-driven horizontal gene transfer can greatly influence bacterial evolution and adaptation to new environments, both inside or outside of a host. Insights into the pace at which microbes evolve within hosts, as well as the dominant evolutionary mechanisms that drive such evolution, are starting to emerge. However, data regarding basic evolutionary mechanisms and, importantly, their effect on the bacterial fitness are very scarce. Animal models with complex or reduced microbiotas offer a prime opportunity to study microbe evolution. Phage-driven horizontal gene transfer is operated by temperate phages that integrate their DNA into the genome of the bacterial host. The integrated phage sequence is called the prophage, and while inserted into the genome is said to be in the lysogenic state, also known as lysogeny. Bacteria that harbor prophages are called lysogens, and the evolutionary value of lysogeny is barely understood in the context of the mammalian gut colonization. Assessing the evolutionary parameters associated with mutation and with lysogeny, including the rates at which lysogens appear in the gut, as well as their fitness advantages and costs, is essential to understand evolution within the gut microbiota. Unveiling the extent of interaction, this is epistasis, between phages and the genomic backgrounds where they integrate, will also be critical to understand the role of temperate phages and mutation in bacterial evolution. In our article, we discuss novel ways and recent discoveries towards that goal. In short, knowing how much bacterial evolution occurs in our intestines and which factors control it are currently key open questions. The formation of new bacterial lineages, ecotypes, is highly likely in our guts. Temperate phage horizontal gene transfer leading to lysogeny is also widespread in the mammalian intestine. Yet, the roles of mutation and especially lysogeny as key drivers of gut bacterial adaptation remain poorly understood. The mammalian gut contains hundreds of bacterial species, each with many strains and ecotypes, whose abundance varies along the lifetime of a host. A continuous high input of mutations and horizontal gene transfer events mediated by temperate phages drives that diversity. Future experiments to study the interaction between mutations that cause bacterial adaptation and lysogenic events with different costs and benefits will be key to understand the dynamic bacterial evolution in the intestine of the mammals. I hope you enjoy the reading, and thank you for listening.